today I'm here with Dr. Stephen Hatfill. He is a virologist, an author, and a bioweapons expert. You probably know his name because in the early 2000s, he was wrongfully accused of the anthrax attacks and won a sizable settlement against the government for that wrongful accusation. Now, he also was known as an early treatment proponent in the first Trump administration, where he was a senior medical advisor. And he was a very controversial figure. And depending on what side of the early treatment fence you are on, you will think of him as a hero. You might think of him as somebody who pushed drugs that you don't think worked. And he's here to clarify that. We have a long conversation about that episode today. And now he is working again in the second Trump administration. He is a senior medical officer and an advisor under RFK Jr. at Health and Human Services. And he's focused on pandemic preparedness. And that's another topic we cover. So just when you think you know everything and all the behind the scenes, you have a conversation like this and you go, oh my God, there is so much we still don't know. Dr. Hatfill clarifies a lot of questions that you had. I took a lot of your questions that you sent to me on social media, and I was able to ask him directly. And I can't tell you what a fascinating, but also shocking and upsetting conversation this was for me in many ways. I just continue to learn about the horrors of the last 5 years, from the virus to the pandemic preparedness plans that we completely ignored, to the early treatment debacle, to harms of the mRNA vaccines, all kinds of things that I think are really important for all of us to know now and continue to learn about. So thanks for joining us, and I think you are going to really enjoy this conversation. Dr. Hatfill, thank you so much for doing this today with me and answering all these amazing questions that I got for you from all kinds of folks around the topic of COVID and biowarfare, which you are an expert on, and pandemic preparedness.
Always a pleasure to talk to you.
You too.
We always have fun on these.
Okay, so I'm just going to fire away, and I'm excited to hear your answers to these questions.
Okay.
What makes it clear to you as a virologist that COVID was bioengineered?
There's a couple of genetic sequences in this virus that we can't establish an evolutionary line from. Seems to be a mix of a couple different COVID viruses. What finally sold me was in the Wuhan strain, the sequence that the US was given.
Mm-hmm.
There's a 20 amino acid stretch up near the sort of the cauliflower part of the protein.
Yep.
That if you three-dimensionally model it, it is a super antigen, which is a bad thing to be. And it's a super antigen that's derived from a bacterium called Staphylococcal enterotoxin B. And what this does is that if you get it into your bloodstream, it turns on about 30% of your lymphocytes all at once. This is what we call the cytokine storm. If you grew the Wuhan virus on human epithelial cells, that segment stayed in there. But if you grew it on alternate tissue culture methods, it chucked that thing out. And this is always sort of an indication it's been crammed in there and it's not natural. And then there was a thing called a furin cleavage site that had been engineered in there. So the spike protein is like a closed umbrella, and it's manufactured, but it's got to get through all that cytoplasm in the cell membrane and poke out through the cell membrane, and then the umbrella opens up, right?
Yeah.
And the furin cleavage site is what holds that umbrella together. And we've seen it in some other coronaviruses, but they're not a direct lineage to the SARS-CoV-2 virus. When this virus came out, it was perfectly adapted to replicate in man.
Yeah.
It didn't like bats anymore. It liked man. And, you don't see this unless it's had extensive passage in a host that is its target.
Does the SEB motif that you just described,
Yeah.
the super antigen?
Yeah.
I read a paper, and I read a paper that you wrote as well, that said, the PNAS paper that I'm referring to originally, it said that motif implies, if it's aerosolized, that it was made as a result of bioweapons research. Do you agree with that
Well,
stage?
it was a bioweapons agent back in the old offensive U.S. program,
Mm,
SEB.
mm-hmm.
And it was designed as an incapacitating agent. In other words, it would kill less than 1% of the population that it was used on.
Mm-hmm.
There were even plans, we were going to spray it on Cuba during the Bay of Pigs as an incapacitating agent because if you breathe it in, it gives you a pneumonia from hell. Seriously. If you eat it, it gives you a terrible gastrointestinal illness. They were going to do another agent as well, and then a third agent after that. And the island would have been sick for a month,
And you think it would have
allowing,
killed about 1% of the population there?
well, nobody did it. We don't have an offensive program anymore.
Mm.
Seriously, in the continental United States, they shut everything down and they built some wonderful research facilities because these new diseases keep cropping up. So we have some labs now totally devoted to new pathogens that no one's ever seen before.
Is the super antigen still in the COVID virus as we know it today?
No, I haven't had time to look at the vaccine sequences,
Mm.
but I'm suspecting in the early vaccines, it was in there.
Really?
Well,
Interesting.
they took this paper was discovered later on after, you know,
Yeah. Yeah. I guess I didn't realize that it was in the vaccine, and I knew it was in the virus, but I didn't realize it was also that SEB like insert was or motif was in the vaccine as well.
I've not seen yay or nay.
Interesting.
Remember, you couldn't get any vaccine to check.
Right.
All they were finding all sorts of weird things. It was only recently, the extraneous DNA fragments and all the junk that's in there
Are you
from
concerned
bad
about that?
laboratory practice. Yeah, I'm concerned about that.
Are you concerned about DNA integration and
Yes, we were by the mid of 2020,
mm,
sorry, by the mid of 2021, they had modeled this and they found four prion sites that could fold abnormally
mm-hmm,
into an insoluble, an amyloid, if you will.
mm-hmm.
And it always had that potential. And again, I haven't seen if that's been engineered out of that sequence.
Interesting. What is the implication of the DNA, the plasmid DNA in the vaccine?
The problem is your cells have a biochemical mechanism called line dash one. And when you see a lot of viral DNA floating around, this is a trigger to your immune system.
Mm-hmm.
It doesn't like that. It doesn't like the methylation on it. And what it will try to do is grab part of it, turn it into, if it's RNA, turn it into DNA, and it ends up going back into the chromosome as a transposon. Transposons are jumping genes. Like maize, like
Mm-hmm.
real corn,
Mm-hmm.
and it's all different colored kernels.
Sure.
That's what gives the different colored kernels.
Ah,
Lady named Barbara McKendrick got a Nobel Prize for finding that. These are segments of DNA that can jump in and be expressed
uh-huh.
and then jump out. But sometimes when they jump in or jump out and re-jump in, they end up next to an important gene involved in cancer.
I see. It does
Okay.
seem like there's a lot of concern about the virus causing cancer and potentially the vaccines being
Well, the
oncogenic.
vaccine, particularly so, one of the ends of the viral genome has something like a SV40 enhancer on it. And this makes certain genes express stronger.
Can you
It's
explain what that is?
kind of like an afterburner for gene expression, like an airplane, all the flames come out. The thing goes faster because you're just pumping fuel into it,
Mm-hmm.
kind of like that, sort of a rocket assist. But it's this random out of the gene, into the gene that and random insertions until you disrupt something that's like a pre-oncogene that's very carefully regulated, like a growth gene. And suddenly you get overexpression of this, things like c-Myc, this type of thing. And we find these abnormalities in a lot of tumors. We're also finding spike protein in the tumors. months after vaccination.
How can we know whether it's from the vaccine or from the virus?
The vaccine has one of the base pairs of the genetic code is a pseudouridine. It's not really uridine, it's pseudouridine
Okay.
because RNA molecules are very, very, very fragile. And there's enough RNase enzymes on your hand. If you're just like working with no gloves and not under a laminar flow thing, you wonder why you don't have any RNA virus left. It all got degraded like on the tinfoil that you were using, the whey things, and it just got chewed up.
Uh-huh.
So to make it resistant to these environmental RNAs, and your body has a lot of them floating around, just looking for stuff like that to chew up. So to make that resistant and prolong its stay inside the cell, making the spike protein, they put an altered base pair in it, and now you find out you can't tear the thing apart. It lasts for at least a couple of years in some people.
Wow. So do we know, have these tests been done to sort out whether it's the spike from the vaccine versus the spike from the
My
virus?
understanding it is this spike from the vaccine.
Wow.
I don't want to talk too much about pre-published material, but studies are now have definitive evidence that this is jumping in to select chromosomes.
What is the propensity for the virus to do any of this aside from the vaccines?
We're not having a dramatic cancer association with just the virus, is my understanding, but we are with the vaccinated people. And you're finding this vaccinated induced spike protein in the cancer cells.
And this has already been published. We can find this information out there because people are going to write to me and say, where can I see this?
There's a few papers out there.
Mm-hmm.
Some are done in tissue culture.
Mm-hmm.
That's an old paper. They'll be able to find that.
Okay.
I think it was done in liver cells,
Mm-hmm.
like really early on, 2021. I could probably haul up a reference, if I go back and look.
Okay, we can do that later.
But yeah. So, and now we've got actual autopsy material that's, being analyzed and stained, and you can see this stuff in the tissues.
If COVID is considered a bioweapon, who would possess the antidote if one exists? And does one exist?
There is nothing for this.
Nothing.
No.
Well, you have been a great proponent of early treatment. So do you still think that that's a valuable tool? And what do you think the value is now in early treatment in terms of mitigating? You're not a person who denies the long-term effects of COVID. You've seen it, you're a doctor, you know all about it. So you're not one of those people that I talk to that says it's everything's the vaccine. I know you acknowledge all things. So what can we do to prevent long COVID? And do you think early treatment works?
Well, early treatment is number one.
Yeah.
And I couldn't believe the pushback against early treatment. You get an infected hangnail, you don't wait until it's gone gangrenous and you have to cut the finger off. You treat it really early. Or a sore throat, or any of this type of thing.
Yeah.
Cancer. Most of this is curable if you treat it early.
Yeah. It's
insane.
The same.
This isn't normal human behavior. These are people that are compromised in some way, either by the drug companies or by their position. Somehow the people shooting for this, they'll get to the end of it. I think the president is going to form a task force or something. I don't know.
Is it true that the EUAs for the vaccines would have not been possible if there was an effective early treatment?
Yeah. Yeah.
What would you use?
Safe, early, effective treatment. Yeah.
What would you use right now today if you got COVID for treatment?
I've had it 5 times.
And and did you get vaccinated?
No.
And how did you do with early treatment?
Well, I kind of exceeded the dose a little bit.
Okay.
But I came down with the Delta strain. I'd been exposed to it before, going into work every day. Mm-hmm. And getting a low dose of this thing is a good thing because you need a minimal infective dose to really get the infection established in you.
Mm-hmm.
If you get below that, you don't get sick, but your body still recognizes it's foreign and you still make an immune response. So you keep getting these little low dose exposures, and eventually you build up an immunity to it
Mm-hmm.
or any of the variants that started to appear.
And the virus has significantly changed from.
Oh, it's constantly changing. That's its survival strategy. Not always, but usually they get milder.
And as you said, the super antigen has been
Yeah.
taken out.
Yeah. So I took a large dose of hydroxy. I was feeling, it was about 3 a.m. My brain quit working. I thought, oh man, my back was killing me, the muscles along either side. And I touched my ears hot. I thought, well, I'm going to go take some aspirin. I
Mm-hmm.
thought I'll just see. And I tried sniffing the aspirin. I couldn't smell it. It's very
Yeah.
pungent. I thought, are you kidding me? Here we go. Delta. And I just followed the hydroxychloroquine protocol, the normal one. But this one, I mean, I was scared of Delta. This
Yeah.
this was a bad one. It had recombined with another virus and became worse.
What did it recombine with?
Another strain.
Oh, another strain of coronavirus.
Yeah.
I see.
Yeah.
Mm.
Sort of like like playing poker, trading cards.
I didn't know that. That's interesting.
Yeah. Yeah. Give me two of these cards and I'll get rid of this one.
Do you think that was bioengineered as well? Was Delta in and of itself a new lab strain?
No, it probably came out of an immunocompromised patient that wasn't able to clear. And there's a lot of variants in these patients, and it was a natural thing. So, I had my own treatment there that I took, and I got up the next morning, I was fine, like no fever, nothing. The smell was still a bit gone. I thought, well, that was weird. So I went in and I got the PCR test, and it came back positive. So I went back a week later, and there were my antibodies. So it was a real test. It was a real positivity. And then I caught a couple Omicrons, like, and you're sick for a day or something with the sniffles.
Did you treat those
Yeah,
infections?
yeah.
Do you think that early treatment mitigates long-term negative outcomes?
The treatment mitigates a lot of things, doesn't
Yeah,
it? Some people were unable to clear the virus completely.
right.
And when you looked at their immune systems, it was like a train wreck. Their immune systems were still being activated. And we're seeing the same thing now. And the even worse problems are if some of these viral fragments, not the whole virus, but fragments of the vaccine are integrated, and it happens to be making like a partial copy of the spike protein, well, that's also going to be antigenic. It's not part of you.
Right.
So now your immune system is under a chronic stimulation.
And that's where we're seeing autoimmunity. We're seeing this
This is my explanation.
because it's so common and
Yeah.
people
Oh, it's a real thing.
yeah, people don't know what to do. Do
Yeah.
you believe or do you know that shedding can occur in individuals
Yeah.
who have been vaccinated
It's
or
it's
infected?
I found it very difficult to believe, but then again, your skin is loaded with Langerhans cells,
Mm-hmm.
not the ones in your pancreas. These are immune presenting cells. You're everywhere. And you saw that Ebola virus could be shed through the skin.
Mm-hmm. Yes.
So yeah, shedding this thing, yeah.
So
It
is
looks
it
like
yeah
it looks like there's some observational studies that show it may be occurring.
in both with both the vaccine and the virus or just one or the other?
I think the virus,
Well, there's questions about whether the spike is shedding from the vaccine as well. Is
I
that
think it's going to
something?
all be dose related. These things were made really badly. You remember that -70 cold chain that they had to be kept under?
I mean, that was never
These are
talked about again
these are
after that
super
initial.
freezers.
Yeah. Yeah, I remember
I
it very
mean,
well.
they'll freeze gasoline.
Yeah.
mean, this is like double compressor things. We keep all our sensitive reagents in there if you're doing molecular biology for long-term storage, and it's a lot easier than liquid nitrogen. Any interruption in that cold chain, and remember these different fats that were used to make the microparticles of the vaccine were forced together. They really don't like to be together.
Mm-hmm.
And when things get warm and thermal energy and fat tends to like melt,
Yeah.
right? Break down into individual fatty acids.
Yeah.
So, that vaccine's been standing a long time at room temperature. You're drawing up a certain amount, and some of those micelles have broken apart, the nanoparticle, and it's soaking up free vaccine messenger RNA. And some lots were manufactured badly. The CDC refused to investigate that, just out and out refused to investigate it. You can see the corruption has spilled over. So you don't know really what dose people were getting. You give somebody an overdose of the polio, and they'll have diarrhea for the next, couple days. It's a virus that it's live, but it's not like dangerous. It's been neutered, but it still affects the epithelial cells. So it's all dependent on dose. And those doses were uncontrolled. If you
Yeah.
take that actual vaccine and you inject it into the tail vein of mice, like in a proportional to body weight thing, these mice get terrible heart damage from myocarditis. So this is why it was to be given into the tissues. But then the CDC says you don't have to withdraw on the plunger. Well, we've been withdrawing on the plungers since syringes were made
Yeah.
to make sure we're not in a big artery or a
Yeah.
big blood vessel and that we're squirting it into the direct circulation. We want it to be in the tissues.
Right.
Well, how are you going to know if you don't pull back on the plunger?
What was the deal with them telling everybody that it stays in the arm, that the vaccine stays in the arm?
That was a lie.
Yeah.
And they knew because when they pulled up the Japanese sued Pfizer, and when they pulled up the bioavailability, you mark the vaccine material. They marked it with a fluorescent dye. So when you dissect the rats, you can shine the light over it and see what's glowing green.
Yeah.
So when they did this study, you could almost read a newspaper under the green light. It had
It was
gone everywhere.
everywhere. Yeah, I remember that study.
20 different organs and tissues. And they lied about it. And the Japanese health ministry actually sued Pfizer to get the data. Within 20 minutes, this thing is already running around your body.
Is it the lipid nanoparticles that are also causing? I know everybody talks about the spike protein being the problem. What's the role of the lipid nanoparticles?
There's polyethylene glycol in that preparation. That's antifreeze. The big thing, I think, with the nanoparticles was its immunoreactivity.
Mm-hmm.
It was triggering allergic reactions in some people. This is why there was always supposedly a nurse around, sort of an anaphylaxis.
Was it also causing direct damage to the heart or to other organs?
Yeah, the vaccine particles are disruptive to cells. The fact that you're getting an unregulated amount and your own body is now rushing in to get rid of this spike protein. And the fact that the spike protein itself is toxic.
Right.
And causing tissue to. So you're making your own poison if you're vaccinated. And all vaccines aren't like this. This was highly experimental. It had never been tried in humans on a large scale before. The clinical trials, they weren't representative of the population at large. There were no old people in it. There were no pregnant females in it.
Right.
They were basically fit, healthy, middle-aged people and younger. There were no babies. It was like you're in the prime of your life. And they had problems with the clinical trials that they glossed over. And, I don't know where the data is. We seem to have misplaced it. Yeah, The law says I have to release all my clinical trial data the minute I get an EUA for use, but I don't want to release it for another 70 years. Do you
Hmm.
remember that
Of course.
debacle?
Of course.
And they got a good judge that says, oh, yeah, you're going to release it now. And then the mask came off.
Yeah.
Now there's all this, how wonderful Peter Marks and these people. Oh, the vaccine, and it's so safe. And Fauci, last month he wrote a paper on how safe and wonderful the vaccine was.
Do you think he took it?
I wouldn't have.
I've heard that a lot of public health officials didn't take it. They got monoclonals instead. I don't know if that's
That
true.
worked. That worked.
Yeah. Would that
I
make
would, I
sense to
would have,
you that
yeah,
that
I
could be
would have
true?
taken monoclonal.
Yeah.
Yeah. A
Yeah.
couple of my friends did.
Yeah, me
And it
too.
works. It works.
They worked really well
But
for
it'll
people I
only
knew.
work temporarily
Yeah.
because the virus, you're selecting for antibody resistance and you're going to get a strain, like rolling the dice. Eventually, if you roll it or pulling the slot machine handle, if you pull that handle enough, you're going to get a jackpot. We just don't know when. And that's how the virus adapts and survives. It's constantly having mutations and just in case something happens to its host. And this is where a lot of our emerging diseases coming out. They're RNA viruses that have that survival strategy.
Mm.
So when your host animal, the numbers start to dwindle, I mean, that's your meal ticket, eh? And you've got enough variants that'll jump out and find a new host animal. And now that host animal is starting to become us. We're seeing about brand new infectious disease that we didn't even know existed, about one a year.
But are we concerned that these are leaking from labs being released? I mean,
Well, the middle of Kansas is a rough place because there's no lab out in the Flint Hills.
but it's easy to go and you take a drone, you do like, there's ways you
There's a lot to that. There's a great deal to aerosol delivery, and especially with viruses.
Mhm.
There's things you have to put in there that, it's still classified, I think. So
You mean to get them to aerosolize and spread?
properly,
Yeah.
these are state-sponsored type things. Any country with a program, I mean, that's the way. You don't poison the water. That's useless.
Mm.
Doesn't work. The way it works, either insects or an aerosol delivery. Or now we've seen a third way, have infected people come in from another country and
They're the vector.
and have a disrupted public health system.
Yeah.
They're the vector, yeah. The White House has Jerry Parker. He's like number one. I know the man. He's super duper. No, we're not going to vaccinate all the chickens. No, we're not going to vaccinate all the cows. Come on, get real. You can't do that. It doesn't work.
People are worried about vaccinated meat with mRNA. That was a question on my list. And since you just mentioned that, is that a concern of yours?
No,
Is there a human health concern in terms of eating
not if not if the meat is cooked.
that
You know,
that would inactivate whatever the mRNA
oh my God,
risks would be?
your stomach acid is about like battery acid.
Mhm,
Okay, it's designed to break things down into their components.
mhm.
If you're working with chickens, like a lot of the infections we've had are people that were actually culling the chickens
Right.
to stop it. So you're in a high threat environment and you don't have the proper respiratory gear.
Mhm,
And you wonder why you got sick.
mhm.
The cows were a little disturbing, but they found that the pasteurization process kills the virus. The deaths that they have were mainly cats because you live on a farm, you have cats, right? They keep the rodent population down. And if it's a dairy farm, you've got milk and you're going to feed the cats the milk.
Mhm.
And they died. And what was kind of worrying, if this thing was going to jump, one would think that it would have already happened.
We're talking about H5N1.
Yeah.
Okay.
Yeah, there's something, I mean, it's been around for quite a while.
Yeah.
If it was really going to go serious pandemic,
Right.
it would have done so by now.
Right.
Not too worried because there's a very good mouse study, 100% lethal strain of bird flu for mice, 100% lethal. You put them on chloroquine, and there's a 75% survival. And when you look at the lung tissues between the untreated and the treated, you got to look pretty close. When you look at the untreated, it's just an atom bomb's gone off in their
Mm.
lungs.
Wow.
So we have a drug out there, but they maligned it so much. Anyway, these experiments need to be repeated, wouldn't you think?
Sure.
And possibly our next flu season, hopefully it's mild, but there'll be an untreated control group and a treated control group. Let's
Yeah.
bring this out into the open. The heart problems in hydroxychloroquine were never caused by the drug.
No.
It was caused by your body's own cytokine response, your body's own inflammatory response against the virus.
Yes.
That was causing the heartbeat abnormalities. And this had been known, but it was a convenient thing to blame on hydroxychloroquine because it slightly prolonged the length of your heartbeat.
Right. This is an
By
old safe drug. I mean, we should just say
uh,
this is such an
by
old
by
safe drug. It's a
by 2020,
WHO essential med.
but in 2021, they actually hooked early COVID patients up to Holter monitors that 24 hours records it. And they gave them the treatment course, and the people got it, and they got over their COVID. And you go back and look, and there's no change in their length of their heartbeat from where they started. It did nothing.
But they
You see,
prevented how many thousands of people from being treated early?
yeah,
It's so
this is the
distressing.
tragedy. The data's there. I wrote a book about this. I
Hmm.
presented the data, but we described exactly what happened in the timeline and who did what and what was wrong and ignored and the whole shebang of why we ended up with a death rate twice that of India, a country with 6 times larger population.
Mm.
They used early drug treatment.
They used a multitude of things from what I remember. I got a lot of contact from Indian doctors telling me they were using all kinds of stuff. They were also using ivermectin and they were combining different drugs. They were using
On,
azithromycin.
on one of the states in India, their state leader, their governor, I guess you'd call him. Okay, everybody's got their drug.
Mhm.
At 12 o'clock lunchtime, everybody's going to take the drug together. We're going to start our course.
Hmm.
And they bang their paces down to almost zero.
Did they use our vaccines?
For India, I don't know. The Chinese had a killed viral preparation that they used.
Don't you think it was interesting that they didn't use mRNA technology in China?
Yeah,
Is it a
yeah,
gene therapy? Can you clarify that?
yeah, it is.
Yeah, I had an ID doctor say to me recently, the only people that deny it's a gene therapy are propagandists. You
Yeah,
look at the
well, the
insert, it's a
CDC
gene.
actually went in and redefined what was a vaccine after 200 years. The clinical trials of the Covaxin were never designed if it could prevent infection,
Hmm. Did
which it
you?
never did. You can't stop a pandemic once it's running
Yeah.
by mass vaccination. You're always going to be behind the virus changing. I thought it would do something, but I didn't realize it was so. I naively assumed these things were well safety tested and effective tested.
Yeah.
It was all kind of crooked.
You had
Um,
concerns as a scientist from before they were released, right? Like just knowing about prior studies prior to 2020, 2019, even.
yeah, I'd written a letter to one of the president's chief of staff,
Mhm.
clearly outlining, look, early drug treatments work. This vaccine is unknown territory. And if there's any failure of this vaccine in any fashion, like safety or efficacy, You don't have a plan B. You got rid of plan B.
Right. Right.
And there you go.
Was this an attempt to perfect the technology? Because now, there's 100 or more mRNA
Oh,
therapies
everybody, yeah,
in
not
the
one of
works.
them. You're correct about that. And they're trying to push a self-amplifying
I saw that.
influenza, the Gates people.
What does that
This has,
mean just for the lay person? What does that mean exactly, the self-amplifying mRNA vaccine, flu vaccine that it is being developed or it's already been developed?
oh, it just keeps making itself.
What does that mean for the human body? Like if you took that, what could happen?
How do you stop it from keep making itself? This is the problem we have with the current vaccine with the
Yeah.
altered nucleotides. It's hard to break down. And you're
Yeah.
right, everything's like mRNA.
There is this deep concern about people are continuing to make spike weeks to months to years after they were
Yeah,
vaccinated.
we're up to 2 years now.
And okay, and also, is the virus doing that? And is there any way to turn it off in either case?
You have to kill it, the virus,
Uh-huh.
either through your immune system or early drug treatment. The beauty about hydroxychloroquine was that it didn't affect the virus at all.
Mm-hmm. Mm-hmm.
It
It was your
changed your
cells.
cells,
Yeah.
it made your cells a little more basic,
Mm-hmm.
and the virus couldn't get out of the little envelope it was in. It
Mm-hmm.
couldn't uncloak to reproduce. Now we find ivermectin has got uses in cancer therapy now. There's a
Yeah.
number of papers out on this,
Very
prostate,
interesting.
breast, this type of thing. So it's gives us a whole new approach now towards what do we need for early drug treatment. We have about 36,000 pharmaceutical compounds that, for one reason or another, never quite made it to the market. People have been going back and doing some look at this towards viruses. They found that a common diuretic agent, a water pill, like
Mm-hmm.
for high blood pressure, kills mononucleosis virus.
Mm.
The kissing disease when you're in
Mm-hmm.
high school,
Sure.
blocks it in its tracks. Epstein-Barr virus. It's also involved in some lymphomas and Hodgkin's
Yep.
disease. A common water tablet. So how expensive would it be to go through these compounds? There's about 220 viruses that cause disease in man. We know to start, probably we want to look at the parasitic agents because that's where we've had the most luck with hydroxychloroquine and ivermectin, two different classes of
Yeah.
drugs, and chloroquine. So I think we've got these national labs. It might be a good idea to start asking questions like, could we do such a study? How much would it cost?
I'm surprised this has not been done in the advent of COVID. I remember Ian Lipkin did an interview in 2020, very early on like CNBC or MSNBC, and he said that we're not following the pandemic plan, where we should be looking at repurposing drugs. And the interviewer asked him why not, and he said because they want something, I'll never forget it, shiny and new and patentable. And it was so disturbing. And I knew Ian had recommended to mutual friends of ours that they take hydroxychloroquine back in that early period of the
Do you
virus.
want to know do you want to know a secret?
Sure.
There was a study, funny enough, published in the Open Journal of American Journal of JAMA Open.
Mm-hmm. Yep.
And they actually had done like telephone interviews and stuff. And they reported that 1 in 20 Americans actually took either hydroxychloroquine or ivermectin for COVID-19. People aren't stupid. And the word of mouth is,
Well, public
it works
officials
faster
were
than
taking it. I know for sure because there were doctors who were saying they were treating senators, they were treating governors, they were
they were treating Congress people.
Congress people. It's
The word of
crazy.
mouth gets out. So what we've seen, this was all self-induced. There's a handful of federal bureaucrats, senior, that the purposes aren't quite clear yet, but they did exactly the wrong thing.
Are you referring to people like Collins, Fauci, Janet Woodcock? Who do you mean specifically, if you're allowed to
Well,
say?
keep going. You're on a good list there.
Yeah. Peter Marks.
Peter Marks, there we go. The senior editor of the New England Journal of
Oh
Medicine,
yeah.
the
Sci
editor of the Lancet, Mr. Horton.
Science magazine. What should happen to them? And are they protected by the PrEP Act from prosecution?
One of them is
Fauci?
Dr. Fauci, I believe it's, unless it was signed by AutoPen.
I don't know, but I'm under the impression that a lot of these people were protected.
I'm under the impression that a lot of those pardons got signed by AutoPen,
Mm,
computer signed.
hmm
I mean, we've got over 1 million dead people. How much was due to incompetence? How much was due to greed, conflicts of interest, this type of thing? How much was it due to these very wealthy drug companies, even more wealthy now, buying people off, influencing the medical journals to publish faulty papers,
Yeah.
showing that hydroxychloroquine didn't work? When you go back and redo the statistics, yeah, it worked.
Surgisphere,
But
some
that's
of
not what
these.
oh, and fraudulent.
Yeah. Absolute fraudulence.
So I think we're talking confinement punishment, i.e., jail.
Will it
If
happen? Like, is it possible, do you think, that anybody will ever pay for what
this
they
is the
did?
United, well, you have to believe in America. There has to be an accountability, and there has to be punishments. Journals that knew they were publishing nonsense, take them off of PubMed for 10 years. See how they do when they're not admitted to the National Library of Medicine.
Mm,
They lost their privilege.
yeah.
They lost their trust. I was really badly shocked when everything I believed in, like the peer-reviewed literature, was just a joke. I could look at the journal and tell you what the article would say without reading the article.
Yeah. Because you
You
knew
go
the
back in,
spin.
you
You knew
read
what the
your
spin
article,
was
and
going to be.
you knew the spin was going to be there, and you knew which journals were doing it.
Mm,
In fact, the editors themselves complained privately to the French health minister.
mm.
They're going to quit publishing clinical trials because, I mean, they're being forced to do it. Well, when you're senior anything, grow a pair. You're supposed to be in charge of things. Resign and make it very public. I'm out of here because these people are forcing us to publish rubbish. And that's racketeering, really.
Do you think that intranasal vaccines are a feasible solution for COVID and
I
for
do.
other diseases?
I do. I do. I do.
Do you think they'll be developed? Like, do you think that, I know there's been some trials, they haven't worked. Flu vaccine didn't work. The inhaled one didn't work so
The flu
well.
vaccines don't work that well anyway.
Yeah, I know. I know.
If you're over 50, forget it. They don't work. Okay, so that's a very good question. Every time you come up with these really good questions,
Thank you.
you don't have just one immune system.
Mhm.
You have site-specific immune systems.
Hmm.
So the immune system that lines your gut is your brain has its own immune system. Your airway has its own immune system. Your lower airway has its own immune system. So if you want to make a vaccine and you're able to get the nasal mucosa, which makes its own kind of special immunoglobulin,
Hmm.
that's a good thing.
So what do you
Yeah.
think in the meantime about intranasal sprays to prevent, post-exposure prophylaxis? People use saline, people use,
They
these
work.
different, yeah.
They reduce the viral load in your upper airway.
Do you ever use stuff like that yourself?
Yeah,
Yeah.
yeah. I'll take a snootful.
Yeah. I don't see any harm. I've been doing it for 5 years and knock
Yeah.
wood so far.
Povidone iodine works very well. Saline, these viruses, they're surrounded by like mucus droplets that kind of keep them. You get it in, okay, now it's in a nice environment.
Hmm.
But all this stuff is very sensitive to the salt concentration of the fluids around it.
Uh-huh.
And you've never done this, but as a kid in Illinois, if we found slugs, we'd go in and get some salt and put it on and
Yeah.
watch it shrivel up. Well, water will follow wherever salt is.
Hmm.
So if you're a virus and everything's really nice, and somebody puts a lot of salt around you, whatever moisture is inside that viral particles, when it wants to be with wants to be with the water. And it kills these things. You got to have a lot of virus in your nose to give it to somebody.
Mm-hmm.
Cough it out. If you're keeping that down with these oropharyngeal, nasopharyngeal washes, you're safer to be around other people and you're giving your body less virus to fight.
So is it possible, do you think, that they can actually prevent, like full on prevent an infection? Or
I don't think,
contain it just in the
I
most
think they can,
minute amounts so it doesn't
think,
disseminate?
I think they can contain it and very possibly keep it, because all you need to do is give it a couple weeks and now your body's fighting back with very specific antibodies.
Mm-hmm.
So if you can drag it out a little bit, plus you're going to feel better, you know. So I'm a big fan of nasopharyngeal washes. It's
Right.
not that bad once you do it the first time.
I think it's totally worth it. I think it's
Yeah.
totally worth it. I don't really see a downside. Do you think that we are seeing vaccine associated enhanced disease or antibody dependent enhancement?
Yeah,
Are you into this
I think it's a
topic?
real thing.
Yeah.
If you look at, and then now it's a little hard to separate, is this ADE, antibody dependent enhancement,
Mhm.
or is it vaccine side effects?
Right.
So the data is all murky now, but we never rebounded as we should have when the milder Omicron strains came out. Our all-cause mortality
Hmm.
is still not normal, and our average lifespan is still decreased.
Do you think this virus is killing in stealth ways? Less of the acute stuff, the immediate cytokine
Yeah,
storm
I think
deaths.
so.
Yeah.
I think so. But this can't be hidden anymore. And every organ we've looked at, there's been problems with the vaccine
Yeah.
because the body teaches you to make its own, to make the viral protein.
Mm.
And the viral protein has physiological effects
Hmm.
on your normal systems. They're damaging. We have males 27 years old that don't make testosterone anymore. The vaccine damaged their gonads.
It's crazy.
We don't know what it's doing to female ovaries,
We
but
know though
we
that
do know
they're finding spike, aren't they finding spike there?
it concentrates there.
Yeah.
We're experiencing infertility a great deal.
Yeah.
Yeah. And we need a study that will go in. I've been working with a group that the next experiment will be to go in. It's a group of gynecologists called ABRG.
Hmm.
I think they've got a website. If you Google abrg.org, their next, after this current study, they're going to start looking at ovarian reserve
Hmm.
in women that were forced to be vaccinated to go to college. We can actually tell how many eggs you have left.
Mm.
We can get yeah, there's graphs that show if we got this level of that, and okay, well, here's how many eggs you have left. And I'm not so sure we're not going to see some horrendous numbers.
How do you, as an expert, and how does the field of experts weigh the benefit of bioweapons research against the potential for global health risks?
The concept of biological warfare, we were the first to get rid of it as a nation. The Japanese tried it's been tried for several thousand years. But the Japanese, they had some success with bubonic plague and insects, fleas, and typhoid for water supplies and this type of thing. This really didn't get going until the start of the 1950s when scientists and military scientists had learned about aerosolization
Mm-hmm.
and really how to do this.
Mm-hmm.
And they did a study where a converted Navy minesweeper sailed 2 miles off the coast of San Francisco, and they released quite a bit of a simulant.
Mm-hmm.
It was a bacteria, but it didn't hurt anyone. They had set up air sampling stations in government buildings all over San Francisco. Now there were no high-rise buildings then.
Mm-hmm.
It wouldn't work as well today. But they released this
Mm-hmm.
as a 2 mile line source. And the winds were onshore and they blew everything silently. Can't even see the cloud. It's
Wow.
all small particle. And it blew it over San Francisco. And when they got the air samplers data, these were designed to breathe air, sample air at the same rate that you breathe.
Mm-hmm.
They found that virtually every individual in San Francisco, some 700,000 at the time, would have received a lethal dose. That was the next fright after nuclear weapons, these technologies.
Are you afraid of that happening to us? Are you afraid of biological attacks? Does it keep you up at night?
We have ways to respond to this.
Yeah.
State-sponsored stuff would be met with nuclear catastrophe. That's well understood by everybody. If you use this stuff with someone with nuclear weapons, you're going to get it.
But I think about COVID being worse than a nuclear bomb in terms of what it did to all of us, the entire globe. Would we even know we were under attack before it was?
Well, how do you stop it?
I don't know. That's the question. It just feels so
Because
inevitable.
it comes back around and gets your own country.
It's true. So that's been the biggest, confusion of all, or that's been the most complex thing to think through. Like, how did this happen? Who did this? And is there a way out? Is there a way out? And is there a way to prevent it? I know that Jay wants to end gain of function, and I appreciate him for doing
I
that.
I
And
I think that's a really good idea. Look at it this way. We're hairless little apes running around, poking our nose. How does this work? Oh, this is interesting. Why does this work? How does that work? And sometimes you get into things that you got the military always looking for weapons.
do you think we can stop this from happening again? Do you think we can stop? You did obviously, I heard when you talked about a little bit about H5N1. Do you feel that we will be able to head off an H5N1 pandemic in
Right now,
humans? Yeah.
hopefully within a couple of years, we'll be, if we get a bad one that's going.
Are you
And
concerned about it emerging, I guess is really my
I
question.
don't like its behavior. If it was going to emerge, it would have emerged by now.
I guess, are you concerned about it being released? There's all this experimentation done on it. And we know Fauci had funded some really crazy experiments with it back in 2000, when he did that op-ed called A Flu Risk Worth Taking about H5N1.
You need to understand, out of all the known
Mhm.
microorganisms,
Mm,
they only manage to weaponize a handful of them.
right,
Being in an aerosol is not a natural state.
right.
You'd think hantavirus would have made a really good BW agent,
Yeah.
the Four Corners agent. Terrible. They couldn't stabilize it in an aerosol, the Russians. It's useless for large area coverage. It's not an easy thing to do. You need a scientific team. You need technical equipment. It isn't something you can hide easily. And there's people that look for this type of who's buying this, who's buying that.
Hmm.
Yeah, a lot of it's dual use.
Yeah.
But, if I'm buying Petri dishes and spray nozzles, hopefully somebody's going to start knocking on my door. So there's people that watch out for this.
Good.
I'm just trying to get our local authorities back to the World War II kind of stage where people were a lot more self-sufficient than they are today and able to handle a pan. Look, if every local authority in the country can manage its requirement for like excess doctors and nurses and extra drugs and extra hospital beds
Mhm.
on the next pandemic, then the country can handle the pandemic. And that leaves their local governments and their state governments and the federal government able to concentrate on other problems in a pandemic, like workforce loss for the
Right.
power plants and the water.
Right.
And our two megacities are so interdependent on each other. If you lose like one thing, it affects everything. So the federal government cannot do everything for everyone at the same time. It was never designed for that. It was to help local authorities help themselves. And you can do this with training and with rehearsal. So when the day comes, if the day comes, everybody knows what to do. They've rehearsed it.
Yeah.
I don't mean sitting around drinking coffee. I mean proper rehearsal.
Mm.
The federal government does this from time to time, but over the years it got very top down, and they forgot to separate pandemic from BW attack. They're
Yeah.
different things.
Yeah.
Pandemics go on for months. BW is over, and I mean, you get your first deaths in 48 hours, and by the end of 2 weeks, it's over, unless it's a highly contagious agent,
Unless it's COVID.
unless it's COVID,
That never
and you
effing
give a
ends.
vaccine that immunosuppresses your body.
Well, I should have asked you, and I did forget one important question, which was the Cleveland Clinic study. I'm sure you saw
Yeah,
this. Well, the study I'm referring to showed that with increased amount of vaccines, I think it was three or more, you had a greater risk of more infections, a greater
yeah,
amount of infections that made you more
yeah,
susceptible. So
yeah.
can you
And
explain?
immunosuppressed you.
Yeah, okay.
Yeah, you do really good against the virus that's circulating there, but they didn't look at cell-mediated immunity. The defense, you have two types, broad types of immunity,
Hmm.
right? You've got humoral immunity, which is, you've never seen this before. You make antibodies against it, and these molecules attack that specific pathogen, and you get rid of it.
Mm.
Then you have cell-mediated immunity. And what happens here is you've got these like killer lymphocytes, and they're clonally programmed to respond to certain molecules. Here comes a new pathogen, and it recognizes it. Oh, yeah, it sends all these chemicals out and recruits the others like it. It's dividing like crazy.
Mm.
And this is like a tank. And now these are all going through your body. And if it sees a cell, one of your cells that are making viral parts, it just kills the whole cell
Mm.
and goes on.
Wow.
There's another one. So it's smashing the factories that are making the cells. By that time, you've had enough time to make antibodies that are circulating.
Hmm.
And the antibodies are like snipers.
Mm.
They're up here and they're taking off the individual viral particles, you see?
Yeah,
And your body clears the virus.
yeah.
Well, it damages the tissue too. The guys that come in, like the tanks, they
Hmm.
damage some of your normal cells too.
Mm.
So, I mean, it's a big mess. It looks like Gaza City right now when these guys have come through. It damages your tissue, but it gets rid of the virus. And it's just buying you time to make these highly specific antibodies that are usually very selective
I see.
at what they take out.
People want to understand what's really happening to them, to themselves, and they want to understand what happened. We're mad. We're all really
Wait till,
mad. We want to know.
wait till you're part of the profession and everything you thought was sacred turned out to be a joke,
Yeah,
including the leadership of your health agencies.
yeah.
They knew these vaccines weren't safe in women, pregnant women. Suddenly you get $11 million and American College of Obstetrics and Gynecology, vaccinate any. Oh, yeah. And here's all these papers that show it's safe. Well, they didn't count the miscarriages, did they? And it
No.
depends which trimester you get your vaccination. You're going to see different effects.
WHO
But
did come out early and say they recommended against vaccinating pregnant women, and then they reneged.
and then they got $11 million from the CDC, but they had to follow the CDC. So they think, well, the CDC have done the study, so it must be okay. No, CDC is in on the take. And I'm still psychologically damaged by all this. You know, you believe in something so strong. I always thought, being a doctor was the best thing I could be.
Mm-hmm.
And you follow the rule and you've got an ethics and you can't be bought and you watch what's going on around you.
Mm-hmm.
And it's just totally disheartening. We've got to put our foot down. This is not behavior that's allowed.
Yeah.
We won't do it again. No, you won't do it again because you're not allowed into the National Library of Medicine.
Yeah,
You had your
absolutely.
chance to do the right thing and you
I think
didn't.
that is a great rule to implement. I would love to see that. I know a
And
lot
if
of
you're going,
scientists who would love to see that as well.
well, if you're going to make an investigation, you've got to match your scientists up with your lawyers because neither one knows anything about the other. So get the guys that have proven themselves, that have sacrificed their careers. Our local doctor here stood up, and that got him a psychological evaluation.
Oh yeah.
He told the school board, like, the truth. And they asked him to leave the meeting and complained, and he had to go be psychologically evaluated.
I think I saw a video of that. Is that possible that I saw that was captured on video? I think I
Yeah,
saw
I
that.
think it was. And there's a lot of cases like that,
Yeah.
more than people realize, where doctors said, I'm doing the right thing.
Mm-hmm. Yeah, and they get kicked out of the
They lose their
establishment.
career.
Yeah.
Yeah,
Thank you so much for your time again. We'll
anytime.
we'll keep talking. Okay.
Okay.
Have a great weekend.
It's good to see you again.
You
Have a good
too.
weekend.
Great to see you. Thanks for joining us today. Please subscribe or follow along for more really interesting conversations. And I'd love to hear from you. So please drop a comment or send me a note. Let me know what you think.